Hello everyone, welcome to MMBS111, Introductory Microbiology. This is mini lecture 8.5, where we're going to be discussing mutations. There are 5 learning outcomes that you need to understand by the end of this mini lecture. First, be able to define mutation. Second, discuss the potential effects of mutations, including positive, negative, and neutral effects. List some of the causes of mutations. Differentiate among the different types of mutations, and finally explain how cells are able to repair mutations. So a genetic change is really the driving force of evolution. Organisms evolve and are able to adapt to their environments because of changes in their genetic information. So a mutation is any change to the nucleotide sequence in the genome. So any change in that genetic code is considered a mutation. Most mutations are most noticeable when a genotype leads, or a genotypic change leads to a phenotypic change. So we have a change in the gene or the genotype, the DNA, and that change results in a change in the trait or the appearance. That's when a mutation is most noticeable. Mutations can involve the loss of base pairs within the DNA sequence, the addition of base pairs, or a rearrangement in the order of those base pairs. And we're going to look at different types of mutation and how they impact that sequence. So a wild type organism is an organism that exhibits the natural non-mutated characteristics. So we consider them wild type for the specific gene that we are investigating. A mutant strain is an organism that shows variants in any of the following characteristics. So they have a different morphology, different appearance in structure or arrangement. They have different nutritional characteristics. They have different genetic control mechanisms. They show resistance to certain chemicals. Their temperature preference has changed, or there is a change to any type of enzymatic function. We would call these mutant strains from the wild type strain. There are several causes for mutations. Some of them are spontaneous, so they just happen randomly, and others can be induced or caused. So a spontaneous mutation can arise from errors during DNA replication, and that's just a random mutation. Nothing caused that, it just happened. Induced mutations create a change in that sequence when the DNA is exposed to certain physical or chemical agents like radiation or toxic chemicals. These are called mutagens, and they are substances that cause mutations. There are several categories and types of mutations, and we're going to talk about each one. A missense mutation is any change in the DNA code that leads to the placement of a different amino acid. And you can see 2 examples of a missense mutation in the image on the left. So the first column shows you what the original strand or sequence looked like. So the original codon was TTC, the mRNA codon was AAG, and this produced lysine as the amino acid. At the end, you can see 2 missense mutations where there was a substitution of a nucleotide, and we ended up with arginine and threonine. So we got a different amino acid than what should have been there. So this is called a missense mutation. A nonsense mutation changes the normal sequence into a stop codon. So you can see an example of a nonsense mutation right here. We had a point mutation at the first codon. It was changed from an A to a U, and this produced a stop codon. So no amino acid is associated with this. This just tells the ribosomes, hey, the translation is done. You've reached the stop codon. A silent mutation alters a base, but it doesn't change the amino acid, so there's no effect from this mutation. And you can see an example here of a silent mutation. The DNA code was TTT, which resulted in an AAA, messenger RNA codon, which also produced lysine. So since the amino acids are the same, there was no effect to the protein by this mutation. A point mutation, like we've sort of pointed out already, is a mutation that changes only a single base within the codon. So it could add a codon, or yes, not a codon, it could add, sorry, a nucleotide, could delete a nucleotide, or could substitute a nucleotide. In all of these examples, these were substitutions. We changed one nucleotide for another. In addition and deletion point mutations, we end up with a frame shift. And this causes a change in the reading frame of the messenger RNA. Remember, the messenger RNA is read in 3 base codons, so 3 groups at a time. Well, if we add a nucleotide or we delete a nucleotide, this shifts how the messenger RNA is read. And these types of mutations nearly always result in a protein that is non-functional. The last type of mutation here is called a back mutation, and this is when a gene that has undergone a mutation actually reverses back to its original base composition. Now, cells have developed a number of ways to repair mutations. DNA has several enzymes that are involved in proofreading that sequence in order to make sure that the nucleotides added are correct. Cells also are able to address things like UV damage. So we have photolyases that actually address and repair damage from light. But they are only successful for a certain kind and number of UV mutations, so they can't repair everything, especially if the damage is pretty widespread across a number of cells. There's also excision repair, so certain enzymes are actually able to break the bonds between the bases and remove the defective bases, and then fill the gap with the appropriate bases for that sequence. So mutations are permanent and they are heritable. This means that once they're there, they are passed on to offspring. Most mutations are not beneficial to the organism. So typically mutations are harmful and they generally end up causing the death of the offspring or the death of the individual. A small number of mutations actually create variant strains that are more readily able to survive and reproduce in their environment, but this is not the norm. In fact, most mutations don't cause any change at all. They're not helpful or harmful. In a stable environment, mutants, so mutant organisms, those that have particular mutations, only make up a very small percentage of the population. When the environment changes, some of those mutants, they'll be better equipped to survive. And drug resistance is an example of one of these mutations. Our use of antibiotics has really selected for organisms that are drug resistant and better equipped to survive that environment. All right, so that's the end of 8.5. Here's a few questions to check in, see what you learned and understand from the mini lecture. And here's a critical thinking question. So using the following DNA sequence, demonstrate a deletion, an insertion, and substitution mutation. Which mutations result in a frame shift? Which result in a missense or nonsense mutation? Are any of the mutations silent? So again, you'll need to use the amino acid wheel from the previous mini lecture so you can determine what amino acids correspond to what codons, and then you can determine what types of mutations were produced by these changes that you made in the DNA sequence. Remember, you can go back and review these mini lectures at any point in time, and hopefully this video was helpful.